李院士，还有熊所长，还有我们今天与会非常多的贵宾，非常多的专家，非常多的学者。那接下来这场 section 我们会有3位 keynote 的 speaker， 那分别会针对这个 clinical trial 跟 precision medicine， 我们来做更多的 elaboration。那第一位 speaker， 我们非常荣幸邀请到的是美国 Stanford 大学非常资深的统计学教授黎院士。那黎院士，我相信在场非常多的学者跟前辈都非常熟悉我们的黎院士。他在1967年在香港大学以最优异的成绩毕业，然后接着到美国的哥伦比亚 University， 在1970年拿到硕士的学位，1971年拿到博士的学位，接下来就担任哥伦比亚的这个 faculty， 从1971年到1974年担任助理教授，1974年到1977年担任副教授。之后，在1986年 move 到这个 Stanford University， 在不同这个 statistic 的 department 担任教授，那也担任 chair。过去20年，对台湾的非常多的我们相关的机构，包括中研院的统计所，包括我们国家卫生研究院都是非常非常重要的这个 consultant 的这个 member。那他今天会跟大家介绍一个 master 的一个 protocol 和 clinical trial， 请大家用最热烈的掌声来欢迎黎院士。
今天我的讲题就是最近我是在 Stanford 跟几个同事，就是 Biomedical 我们的团队一起去做这个问题，就是 master protocol， 就是这个一个方案。我们呢，就是精准医学里面就是主要我们的结果这个我在这个 Stanford 就是里面这个就是在 Stanford 的 Cancer Institute 里面，就是这个我的他的Swami Kumar 是Professor Buster 是那边的医师。那么我跟我同事录影，我们是就是在那边我们负责就是上过统计的工作。那么聊天是我一个博士生，所以其实我就是我们最近这几年就讨论这个问题，然后我们其实有一个药 ，OK， 就是就是那个药本来是一个台湾的一个公司研发的，那么所以结果呢，最后呢就是会就是我们的药。那么主要我的这方面，这个我的国语就是不太流利，就是尤其讲到这些名词，所以我就是以下因为这时间的缘故，我就是会讲，我就 switch 这个就是另一个。So today, so the outline of today's talk means master protocols to study multiple therapies, multiple diseases of both. And actually there is an article by Janet Woodcock, who is the director of CDR at the FDA. And Lisa Lavanche, okay, was the head of in the biostatistics group, okay, working under Janet. But and others, but then Lisa is also the president of the the current president of the American Statistical Association, and she has recently resigned from the FDA, and she'll be moving to North Carolina State. Okay. So they wrote a paper, a recent paper, in 2017 in the New England Journal of Medicine. And it talks about mechanism-based, so this is where the precision medicine comes in, okay, because in this age of precision medicine, and actually when we talk about cancer trials, okay, some of these, okay, we'll see how it comes in. And the idea is to precision medicine trials, and the reason why, they advocate, because precision medicine trials, in principle, if we use the old paradigm, will be very expensive. but now here is affordable, okay, in cost, time, and also sample size. And then there are several types of these massive particles. And actually in the paper there is 6 such trials, okay. And so I will explain, okay, I give examples of, they are the platform, umbrella, and basket trial. Now, so this is really, something, okay, that Janet would talk, okay, who also visited Stanford, okay, in April, and I talked to her about this. And she thinks this is, actually one of the, things can break this lockdown, okay, because for a breakthrough, okay, if it is conducted properly, if one has innovations, real innovations, then we can resolve the issue of these very expensive trials, okay, in the confirmatory phase. So there are challenges, obviously, in design and analysis and basically I will focus on basket trials, although there are also parts of the other trials. And what I want to, we have been doing, especially with the trial, okay, that I mentioned about, okay, that we are really working on, okay, is an adaptive design and using innovations, statistical methodology illustrated by one such trial currently planned at Stanford. So, these are the new 2017 paper of Wilcock and Lagrange. So they list, 6 such trials, okay, with master particles. So, and then I sort of reorganized the table, and so because I want to talk about basket trials, it's today's theme. So, so these are the current the trials, okay. They cite, okay, so there's B225, so that's the name of the trial, but I will basically focus on BRAF V600, okay, which is an early phase 2 trial. And then the NCI match, okay, it's an umbrella trial, okay, and its design is basically an exploratory proof of concept design. And then there is the battle the battle trial, which is an umbrella trial, in phase 2 design as a phase 2 trial. And then there is this famous I-SPY 2 trial, which is a platform trial, a phase 2 trial. Then there is a lung map, okay, which I will explain at the bottom, is a phase 2 to 3 trial. So what is a basket trial? So in principle, basket trial is the study of a single therapy. So I told you this company, okay, actually headquartered in Taiwan, and it has developed a therapy, a drug, and but it wants to use it for multiple diseases or disease subtypes. So that is basket. Umbrella is that you now study many therapies, okay, but now you want to use them for a single disease. So so that's umbrella. Now here you have a single disease, and then you have now what is platform. And platform is like an umbrella, but then you have these therapies. Some enter the trial, and others exit from the platform. So there's entrance, and exit. So I-SPY 2 is such a trial, okay, because and now there's the lung map. Actually, is something, okay. In between, it is used to study 3 drugs. So instead of a single drug, okay, single as a basket, okay, you have 3 drugs. And so now you have multiple therapies, and it is now basically in phase 3 for non-small. But now actually here, in phase 3, but then there are 4 molecular targets initially, and then trimmed to 3. So basically here, it treats the lung map is multiple drugs, multiple disease subtypes. So that's the now here, because I want to focus on the BRAF V600 mutations occur in about 50% of cutaneous melanomas and result in constitutive activation of downstream signaling through the MAPK pathway. Based and now here the scientific background articles in Nature 2002 and New Journal of Medicine 2005. And now here is the drug, okay, which is developed by Roche Genentech, okay, and Velurafenib, okay, is a selective oral inhibitor of BRAF V600 kinase, and is associated with response rate of about 50% and improved survivals with BRAF V. Now there is an earlier study, so it is targeted to melanoma. Okay, so mutation positive metastatic melanoma. But now actually they want to use it. This BRAF V600E is essentially just usual. Okay, it's not a massive protocol. But this drug, Hyman, wants to use it, for different, as we say, different kinds of cancers, not just melanoma. So efforts by the Cancer Genome Atlas and other initiatives have identified BRAF V600 mutations in non-melanoma cancers. Okay, so first, people have used this, and of course these are small studies, phase 1, phase 2 studies, that sort of think this will be useful to treat it. So that's where the conclusion that the efficacy of what they want is the Roche genetic drug. In these non-melanoma cancers has not been systematically explored despite its therapeutic potential. So this is a quote from this Hyman's paper. So that's the basket trial. The large number of tumor types, low frequency of BRAF V600 mutations, and the variety of some of the non-melanoma cancers make disease-specific studies difficult, unaffordable. Is my word. So to conduct, because we have so many diseases that see that this drug, that has been approved, has promise. But now, to really conduct it for each of these, is not, is unaffordable. So the idea is, this Hyman sort of conducted the study with 6 baskets. So these are the cancer types, not just melanoma, but now here, non-small cell lung cancer, ovarian, colorectal, and breast, multiple myeloma, and cholangiocarcinoma, and the 7th. So these are the specific baskets. They think this drug can be useful. But then, since it's a basket trial, so there may be others, that are admitted to this study. For the patients, so they put a basket, so-called all others, the 7th basket, which permitted enrollment of patients with any other BRAF V600 mutation positive cancer. So that's the goal of this study. So we have phase 2 study, not yet confirmatory, but so the goal of this study was to identify promising signals of activity in individual tumor types that could then be definitively explored. So now here is so what design? How is it done? So what it does is to use as with all kinds as with this kind of phase 2 studies. So it's an adaptive Simon 2-stage design. Which is basically there's a 2-stage. So initial, you sort of look at it, make a go/no-go decision, and then if you go, then you go to the end. If no go, then you stop for futility. So this is a Simon 2-stage design, and it was used for all tumor-specific cohorts. Okay, instead of just one specific kind of cancer, is what they list, tumor-specific, in order to minimize the number of patients treated was deemed ineffective for a specific tumor type. So the primary efficacy endpoint was response rate at week 5. Kaplan-Meier methods were used to estimate progression-free and overall survival. The adjustments were made for multiple hypothesis testing that could result in positive findings. Okay, so that basically, because of all these tumor subtypes, essentially there are multiple tests, but they haven't adjusted this. Okay, so and now at the end, okay, so this trial they had 122 adults receive at least one dose of the drug, and now here is the distribution: 20 for non-small cell lung cancer, 37 for colorectal cancer, 35 for multiple myeloma, and 8 for colorectal carcinoma, and 18 for another kind of cancer, ECD or LCH, okay, something. This is what I label as all others. Originally it wasn't in the basket, but it turns out, okay, there are quite a number of those, and 34 for breast, ovarian, and other cancers. Okay, so other actually ECD or LCH was there, but actually there is quite a number following that. So 89% of these patients had at least one previous line of therapy. So that's basically a second line therapy. And so the drug, okay, showed efficacy in brown meat, so that's the results. And B600 mutation positive non-small cell lung cancer compared to standard second-line docetaxel in molecularly unselected patients. And now actually what happened is for this all others, it turns out for ECD or LCH, which are closely related orphan diseases. Now, orphan diseases means that they actually there are no drugs, okay, with no approved therapies for them. Okay, so that's why they're called orphan diseases. So actually, it turns out the response rate was 43%, and none of the patients had disease progression while receiving the therapy, despite a median treatment duration of 5.9 months. So, so that's the kind of things, okay, so this drug, okay, look, now they have, so I say basket is one drug for multiple diseases and multiple disease subtypes, and actually it showed, okay, it showed very promising results. So one challenge in interpreting the results of basket studies is drawing inferences from small numbers of patients. So I have shown you before, that's the number of patients, okay, 122, but then, okay, actually you spread them out, okay, over the different diseases, so actually you don't have that many to talk about. So are these results, and also there is the issue of multiple testing, and so what is the significance of these results? So that's and actually Don Berry, okay, who I talked about I-SPY 2, was actually, he was the one, he's at MD Anderson, okay, Texas, with the Cancer Center near Houston, and he sort of wrote a paper, on these trials. So he discusses other challenges in inference from basket trials in particular. So these are his comments. Okay, of course he was advocating Bayesian. Okay, in fact all these when we talk about adaptive design, so Berry is one of these pioneers, and actually is still very active in the area. But all his strategies are Bayesian strategies. So he discusses other challenges in inference from basket trials. So and here are just the major ones. Even though patients have, the same biomarker, different tumor sites and tumor types may have different response rates, and simply pooling trial results across tumor types may mislead interpretation. On the other hand, different tumor types may have similar response rates and hierarchical, so that's what he was basically proposing. I mean, here you say here, so he uses hierarchical Bayesian modeling, and so to borrow information across. So why do you do a basket? So he did that in mind. Okay, so there's some sort of commonality. So hopefully you can borrow information across baskets, and that is using a hierarchical Bayesian model, and it's a way out. Okay, so that's at least okay to do posterior inference. Now, so that is his point. Now I'm currently writing a book, okay, which should come out next month, okay, with Joe Hayes and Jay Chen at Merck. And our book is on so medical product safety evaluation. So in other words, you have different drugs or medical devices, and you want to evaluate their safety. Okay. Now the drugs, okay, presumably have been approved for efficacy, and they haven't shown serious adverse events. Okay, in these pivotal clinical trials. But then maybe when it is put in use in the market, I mean, some adverse events and so on will show up. So, I mean, so we discussed in our book, okay, so we discussed the connections between frequencies and error controlling methods, and Bayesian, in particular, empirical Bayes, which is something that we favor more than at least we use the data to determine, okay, this prior model. Methods to address multiplicity issues in the analysis of clinical trials data. So, I mean, for safety events, because for safety, now you actually, there are a lot, as I say, a lot of unexpected adverse events. So are they really there, okay, or are they just so that's the issue. So we focus in our book on evaluation of adverse outcomes of a new drug medical product, which seems to be unrelated to this basket, but the statistical problems are quite similar. So the reason is adverse event types, okay, so usually when you report, I mean, so there are definitely dictionaries, okay, that are used, okay, I mean, to report these adverse events, okay. And then the dictionaries, okay, have a certain hierarchical structure. So you talk about now, because the different parts of the body, okay, may be related, so what part of the body, okay, has it? So the hierarchical structure, you talk about the same body, when you talk about adverse events, you say which body system or organ. So now, if you look at the analogy with basket trials, with precision medicine, is genetic aberrations in organs under different baskets or cancers. Okay, so you have again, okay, it's this body system or organ. I mean, it's just a basket, different kinds of. But what is really, I mean, these adverse events correspond to the genetic aberrations. And then here, when we talk about the domain knowledge about these drugs, okay, or something, it's the biological models of toxicity. When we talk about safety, and here, when we talk about precision medicine, are the mechanism-based background of treatment and so on. So now, so here there are issues, okay, statistical issues that Barry has raised, and also, and even in Hanum has also raised, okay, in his in the Living Medicine paper, but there are similarities, okay. And so in our book we talk about, and actually, I mean, when we talk about Bayes, frequencies, and all of these, in fact, the multiplicity problem statistically is basically a multiple decision problem. So dating back to Robbins and Stein, and now recently Brett Efron, we use it for microarray and gene expression analysis and so on. So they are compound statistical decision theory for optimal balance between type 1 error and power. So, and its relation to empirical Bayes approach. So that's okay. So now here challenges. So I will speed up because I'm going to. So I just give the background of this foundational paper of hype. But and now where do we come in? So I talk about Swati Kumar. So she was originally at the NCI, okay, and then she joined us, okay, at the Kansas Institute a few years ago. So now before she joined us, okay, because I told you her background was at the NCI. So when she came, okay, she was a network of investigative research. And actually at Stanford, okay, there is now, before I talk about the BRAF 600 mutation, I mean, that's related, okay, to the genetic drug. But here, there is that's also a direction of biotech. Because these days, a lot of these new innovations, okay, I mean, the drug companies, because it's a lot of risk, okay, when you invest in these developments. So these are a lot of startups, I mean, small biotech firms. And actually the one that she had collaborated is called Luxo Oncology. And this at Luxo Oncology in South San Francisco, and also she had a network of investigators, so through the NCI, and collaborators at UCLA, UCSC, Harvard, Cornell, NW, MD Anderson, and so on. So they designed and carried out a basket trial involving 7 specified cancer types and 8 basket other cancers to evaluate the efficacy and safety of, this another drug, okay, which is a highly selective TLK inhibitor produced by Luxo Oncology in South San Francisco. And now here, and what is interesting in this is in adults and children who have TLK fusion positive cancers. So now this drug, they enroll only, so far 55 patients were enrolled in one of 3 protocols and treated with this drug in a phase 1 study involving adults. Now actually there are several studies. One is a phase 1 involving adults, another is a phase 1/2 involving adults and children, and another is a phase 2 study involving adolescents and adults with TLK fusion positive tumors. And the phase 2 study uses the recommended dose of the drug twice daily, and so on. And it's also because of phase 1/2, you also do the dose escalation, and so on and so forth. So anyway, so that's the primary endpoint, okay. And now, and the data cutoff date of July 17th of last year, the overall response rate was quite impressive, was 75%, and 7 of the patients had complete response, and 34 had partial response. So this was reported this year in the New England Journal of Medicine. And in the accompanying editorial of that issue, Andre, okay, says that this study is an illustration of what is likely to be the future drug development in rare genomic entities. And then according to the magnitude of clinical benefit scale for single-arm trials recently developed by the European Society of Medical Oncology, studies that show rates of objective response of more than 60% and a median progression-free survival of more than 6 months, as the study conducted by Drone et al. Thus, are considered to have the highest magnitude of clinical benefit, in line with the pathway for single-arm trials of treatments of rare diseases. Notice I talk about if they are adults and they are children, okay, and these for children, okay, these are rare diseases and also well established. So natural histories to receive approval from regulatory agencies. So it mentions, okay, that the study did not find any difference in efficacy among the 12 tumors histotypes, proving a successful trans-tumor approach in the case of TLK fusions, but then some basket trials have not shown evidence of trans-tumor efficacy of targeted therapies, notably BRAF inhibitors. The concept importance of developing statistical tools to support the claim that the drug works across tumor types and to provide more in-depth understanding of the failure of some targets in trans-tumor approach. So basically, this is why how we got Swati study about this drug. And that is somewhat, different and quite similar. And actually this was by Teresa Persner, our medical collaborator. So there, so this is what I talk about, and actually about this one company, which is also like a Locke's oncology. It is sort of a new startup kind of company, separate. So this you want to. So here, it produces a potent and selective inhibitor of CK2. Kinase 2, that's what CK2 stands for treatment of medulloblastoma, MB, which is a pediatric brain cancer. So that's the, so here, for 2. So the drug is believed to be efficacious for other cancers, not only for this. So that is what the company wants to market, at least several indications, believed to be efficacious for other cancers. They are associated with PETROP, so the biology is this PETROP signaling pathway. Hence, an adaptive design of a basket trial is used. And the baskets include MB and myelocosarcoma for children, and basal cell carcinoma, so BCC. So these are the two kind of things, colon, pancreatic cancers for adults, but so BCC and MP are what the two indications that the company wants to market. And now, of course, they want to look at other cancers. And actually, what happens is that Teresa Persner wants to look at certain, certain cancers for children. These are the so-called orphan diseases. They don't have approved treatments, and also they are rare diseases. So that's what this company says. Agrees. To give us the drug, the cancer. So this is a basket of other cancers. So that's the basket design. But here it is a little bit different. So the company's plan is, essentially because it has a marketing aim. So the first, here the difference between this and all the others that I've talked about is that the company at this moment wants to focus on MB and BCC indications. So they have already designed phase 2 trials, and now so we sort of work develop some adaptive design instead of Simon's 2-stage. So we use some adaptive. So work out with Bartroff, who was my previous postdoc, and USC, and Rachel Shi was my previous student, and currently at the BA. So at least one of these that proceed to the basket trial. So this is the basic kind of things that we work on, and now since I was the time allowed, so I will now. So I will now go through. So Woodcock and the Barnes say, so now back to Woodcock. We have multiple questions to address under a single protocol, usually in an area of unmet need, and an extensive infrastructure in place to handle data flow. Master protocols are natural environment for considering innovative trial design. The flexibility to allow promising new therapies to enter and poor performing therapies to discontinue usually requires some form of adaptive design. But the level of complexity of those adaptations can vary according to the objective of the master protocol. So our recent work on adaptive design basket trial for CK2 inhibitor illustrates both new challenges and opportunities for adaptive design noted by Woodcock and Lamarche, especially how the design should adapt to the objective of the master protocol during the course of the trial. So thank you.